Ja, ich bin ja so froh, dass der...
Thank you very much for coming back, and it's my great pleasure now to guide you through the first session entitled Personalized Medicine. So if you're looking for free places, we have plenty of them in the first two rows. Don't be shy. And I'd like to share some first thoughts, and then we continue with, I think, great presentations. So throughout the day, our esteemed speakers will share their expertise, their experiences, and their visions for the future with us. From groundbreaking research findings to innovative cloud-based solutions, we'll see the many different ways in which digital health is already having a measurable impact on us. The overarching theme of today's discussion centers around personalized medicine. So it will bring a paradigm shift of what we recognize for the individual solutions of our patients of today. So we believe that big data, cloud computing, and AI are the cornerstones on which personalized medicine will further develop, and they provide us with the tools to decode the complexity of human health in ways previously impossible. And with this, it is my great pleasure, and I'm honored to now introduce Professor Thorsten Haferlach. He is the CEO, head of the Munich Leukemia Laboratory. He is a clinician by training, and we're happy to have him today. So the title he'll talk about is Artificial Intelligence in Hematological Diagnostics, Game Changer or Gadget. Thank you very much for coming.
Thank you. Thank you very much. Thank you very much, Moritz, Dr. Wieler. Thank you very much for the invitation and kind introduction. I'm a hematologist. I'm no pathologist. I'm no lab doctor. I'm no AI specialist. I just speak a little bit about what I do and what this title might mean to you as well. I have to disclose that I'm one of the part owners of the Munich Leukemia Laboratory. It's just next to the Klinikum Großhadern. And these are the methods and the readouts and the phenotypes that we have to apply to diagnose leukemias and lymphomas as quick as possible, to start with the more and more personalized treatment approaches. And I will show you some of the examples how these methods are now used in pilots or in routine and supported by AI tools. So there are a lot of reasons to confuse phenotypically driven machine learning models here or there or here. And I can tell you that it takes some time to really driven, like cytomorphology or chromosomes, is a very good tool to be trained with such models for AI in routine, but you need data, and data. So we start with the very first step of diagnosing leukemias. It's the peripheral blood differential count. This is a blood smear. A completely automated microscope takes these pictures from the smear, takes several magnifications, and finally the readout. And this is now not a pilot anymore. It's in our routine since 1 year, having investigated more than 35,000 cases in routine. After the staining of the smear, these stained smear goes to the microscope, the pictures are taken completely automated, they go to the cloud goes with the AI system, looks into this first time. How does it look like? This is what the microscope is doing, day and night, 7 days a week, never ill, no weekends, no holidays, and this is the lower magnification. The picture of the respective white blood cell is in the middle, and then the oil is automatically pipetted on this microscope, and then the higher magnification, these are the respective cells. And we did a pilot, nearly 30,000 cases blinded. On the left side, 3 million cells have been differentiated and counted by technicians and hematologists, and finally led to the result, leukemia. What other methods are needed? No leukemia, healthy. And in parallel, 5 times as much cells have been taken as pictures. And this is on the left side, the humans, on the right side, the AI, the percentage of these differential count. I can tell you that the concordance, especially of the critical cells, is above 94%. And this is also true now, not only for this pilot, but also for our routine use. And I can tell you, if you delay on phenotypes, it's never yes or no, 0 or 1, because it's a maturation of cells during the line from very immature to very mature. And even me, doing this now since nearly 40 years, I have never the same above most humans that do this job worldwide. And the same is true for another technique. It's called immunophenotyping, where you use antibodies that are fluorescence dyes linked, and we can use this now, but not with phenotypical pictures like scatter diagrams we normally have as a readout after compensation and gating from personnel and machines, but we take the matrix of the raw data. CD3 expression 10%, CD35 expression 0%, and so on and so on. And this raw data, this matrix, is going to the AI tool, and then finally you receive an information. There is no evidence of MDS, it's a malignant disease, with a probability of 99.36%. And these pictures are calculated from the AI back to your eye to control the result. But this AI doesn't need these pictures anymore. They just rely on the raw data of the matrix. This is quite helpful now in our routine, and we are also just moving from the pilots to the routine use. AI first, human next, then the report, and then the results. So this would be the layout and the readout for our human approaches, and everyone is doing this worldwide. And this is the readout and the layout after the AI had worked. And here are the pathogenic cells, and the rest are no pathogenic cells. Just to give you an idea how that works in parallel to our routine setting, and more now supporting our routine settings. Now, this is one of the most interesting examples. This is a metaphase. You may have all done this in your human genetics practicum, in your studying. You cut it with a scissor, then you put the chromosomes on the right order. So this is now all taken digitally by the same microscope I showed you before for the peripheral blood differential. This is the metaphase. And now here, from this moment on, one of our very experienced technicians has to go from the metaphase to the karyogram. My karyogram finally will lead to 46 chromosomes XY. The others would be XX, woman. And she has to cut these chromosomes one by one because the machine doesn't really see it's 2. They thought it's 1. After that, these chromosomes are ordered digitally by the system only by size. And then by phenotypical knowledge, she knows this is a 1, this is a 2, this is a 3. We have always 1, 2, 1 chromosomes to 2 chromosomes, one of the father, one of the mother. And then you see, it's real time, it's quite quick, and you need to know what you do. So normally it takes minutes, or I would need days to do something like that. Maybe I shouldn't try that to do that, but this woman, this technician works with us since 15 years. So we all over have pictures from 300 million chromosomes ordered from a metaphase to a karyogram. And now she's ready. It's a man, XY, down here. And this metaphase has a trisomy 8. This is human done, and this is very, very quick. It took 48 seconds while I was speaking. Now the same metaphase, we start here, and this is all AI. You see, no one is doing it. It's just AI. It's all AI, and especially this is also all AI. And then the human control starts. And the final, including human control, is 12 seconds compared to 48 seconds, and 48 seconds was quite quick. So we completely retrained our personnel, especially the new people have a new training now. They should not start with what we have done 40 years ago. And now this is the approach. So you take the pictures by a completely automated microscope. I show you now 20 metaphases, and this is the real speed, how the metaphase goes to a karyogram. Here we go. 8,000 metaphases an hour before human control starts. You see, this is really a game changer. That's not a gadget. We changed that now after 3 years, in routine, and I can tell you that the turnaround time goes down from 7 days to 5 days for the final result. And the personnel increases because we have more samples, but it does not increase parallel to the number of samples, but it's much less personnel in comparison to samples. From chromosomes to genes. We heard about genes, we heard about genomes. I'll come to back this later. So now we study for specific kinds of leukemias all these genes for myeloid diseases and all these genes for lymphatic diseases. This is the hotspots. We know that these genes are 1, 2, 3, 6 of them, 10 of them involved in specific malignant leukemias and lymphomas. We heard about that Google tool, and we have the raw data from the next generation sequencing machine. We have the mapping to the human genome, and then we have all these variant discovery callers. That was the cooperation with the Mayo Clinic we heard before in the keynote lecture. And then, and this is the most important step, and this is already then driven by human knowledge, variant interpretation. And here we implement also an AI-based tool. And this tool gives you automatically all the other tools that are available and the knowledge about this specific mutation. And it gives you a specific predictor. How often did we see this mutation in our data set? And what was our final decision by bioinformatics and by molecular biologists and by all the other methods in parallel? And this is automatically given into this AI tool and helps us to interpret the next and the next and the next finding of the chromosomes and especially these molecular changes. Is this in routine? Yes. For these panels, it is. For genomes, it is on the way to be that. Do we think it's feasible? Yes, we do. And this was a paper published by the colleagues from the Washington University in St. Louis, and they said, and this is true for us as well, from the patient's blood or bone marrow to the final report of the genome takes around 5 to 7 days. And the Americans are quite quick with that. So this is the NCCN guidelines, how to diagnose acute myeloid leukemias, that was published in March this year. You see all the other techniques I showed you in part before. And after that, they write our whole genome. We are far away from that in Germany, but we are on the way to that in the world. How does leukemias look like? So this is a specific kind of leukemia, but this is 107 patients. All the DNA overlaid of these 107. All these patients have a translocation between chromosome 9 and 22. Here's chromosome 1, here's 22, X and Y, gains and losses, translocations, gene mutations. This is MPN, a little bit more critical and more active disease with respect to chromosomal or molecular changes. Chronic lymphocytic leukemia, MDS, AML, B lineage or T lineage ALL. And there you see it's not just the phenotype, it's not just the cell on the blood smear. It's a very, very complicated for the diagnosis, for the classification, for the prognosis, for the precision medicine, and for the follow-up studies for measurable residual disease. And you are not able in your own brain to organize all that without AI. So we use all our knowledge of more than 6,000 genomes and transcriptomes from our dataset in the cloud, AWS cloud in Frankfurt, according to all German and European data security laws. And the dataset is 4 petabyte big. You cannot store it on your laptop. 4 petabyte is a Netflix video download 100 years in front of your television screen, day and night. This is 4 petabyte. And therefore, if you want to work with this data and to store that data, you need cloud infrastructure. And there is cloud infrastructure in Germany, and there is no problem to do that. So using that and using our gold standard diagnostic approaches with all the methods that we apply today, according to rules, according to classification, and in parallel, using the genome and the transcriptome, the accuracy to predict the right diagnosis now is 88% without all the gold standard methods, just based on the genome and the transcriptomes. So we are moving forward from phenotypically method plus method plus method usage to genomes and transcriptomes in the future. And we have this explainable, we have this transparent. So this tool that we use, driven by AI in-house here, tells us why this is this disease, gives us an information how sure we are that this is this disease and why it was called this disease. And every information is given here, including the DNA and the circus plots, what is on that genome on the DNA level, and the probability that this is a chronic myeloid leukemia with a probability of 97.12%, and the second best choice and the third best choice as well. Now, large language models, ChatGPT, you know all that. We use that as well. We have now more than 1.1 million cases, completely digital. We use half of them, 500,000, and have the question of the doctor and have the final result, and we have all methods in between and all the results digital. So we train the model and say, well, these are the historical data, this is what we did, or the methods, this is what we wrote. This was the final report. Now, this is the new case. Here's the question, here are the results, and match according to our rules, and write the report before a person looks into this report. So we are quite nice now in the area of 86% correctly written reports before a human looks into the report. These 14% are sometimes because the human made a mistake, not the AI made the mistake. And if we don't go into detail, if you want to see reports like that, one above, the other down, and you ask the question, who was the author? This was AI and this was the human. So it is before the human starts to think, bring AI to help you to support what you have to do as quick as possible to support leukemia diagnostics. But, and we heard a lot of that in the talks before as well, there's much more than any kind of laboratory results, RNA, DNA based. There's family history, there's cognition, state of mind, anything like gut microbiome, very important. So all this can also go into this deep learning methods and go finally to really describe what the patient looks like. I like this book a lot. It's from Eric Topol, an American human geneticist and cardiologist, and he wrote, AI will not replace physicians. However, physicians who use AI will replace those who don't. And I think this is a very important message, especially for the young people in the room. What I do and what we do and what we will do not only for in-house, but also as an FDA approvable tool for others who want to upload their data. We have the question, leukemia, lymphoma, you can ask any question, cardiac disease, you have the question. You have the more or less readable paperwork that comes because the doctor wrote what he wants to know. Then you have an order control. What methods are applicable for this specific question? So you have the complete team that is doing something. And finally, you have the personnel, and then you write the reports, and then you include by large language models other information from PubMed, from UpToDate, from NCCN, from clinical trials, patients. And all these steps can be supported and will be supported or are already supported by AI. This is what I see right now when we think about leukemia diagnostics as a blueprint for many other options in medicine in the future. I would like to thank Claudia, my wife, for cytogenetics and AI, Wolfgang for flow and AI, Manja Magnhofer for molecular genetics and AI, and 3 of our AI specialists, bioinformaticians, and the complete team working in routine and cutting-edge technologies. Thank you very much for your attention.
This was remarkable. Thank you very much for this wonderful talk. I think we all agree it's not a gadget, it's a game changer already in use. Are there questions? Yes, please.
Hello, thank you for your talk. It was really interesting. I have 2 questions. So my first question is regarding your large language model. You say you have an accuracy of 86%. Are you trying to improve this? And also I saw you use ChatGPT 3.5. There's also 4.0 at the moment, so maybe this would also help. And then my other question is, you had this slide with all the genes listed on there. And I'm wondering, where are the genes from? Did you use them from databases? If yes, which database? Yeah.
So for sure, we are now on GPT 4.0 in-house, not there. We can implement it in-house and use it there. We are now not 86%, we are already approaching 90%. And again, some things are not quite different. They are not 100% the same, but they are not wrong. And sometimes doctors were wrong. So that is improving quite quickly, and we have all this data there and all these reports there. The genes come from everything that we also do, research, papers, peer-reviewed papers, and WHO classification, where all these genes are known to be combined with specific diseases, and therefore these genes come there.
So you do like a systematic review and meta-analysis?
We do this every day.
Oh, okay. Thank you.
This is our business.
Professor Schmidt, please.
Hi, very nice talk, very impressive. Next step would be to go to a more precision therapy.
Right.
I mean, single gene-based therapies, targeting driver genes is one way, but not the solution. So how do you go further?
No, absolutely true. So we are doing that already when there is a specific diagnosis, a specific gene, a specific translocation in the specific gene, and we already do this since decades, write this into the report. We are not supporting Novartis, Roche, or any other company. So we just say this is the name of the generic of the drug, but not the name of the prescription, because the drugs can have different names. We are doing that already. It's getting more and more precise now, and this is what we also do, because we know that the doctors can't follow all these genes, all these drugs. They have so much colon cancer in comparison to leukemias that they need to know more about colon cancer or prostate cancer or breast cancer in comparison to leukemias, that it's also our important contribution to writing the reports to also give an idea what studies are available, what drugs are available, and where you go first. And this is one of the reasons we would like to add this information by a large language model to the final report. These are the information, DNA-based. Here's what is available right now. Link to this information not only by paper. In Germany, you're allowed to fax the reports. So, but this is the way we have to go. Link it to the real world data and knowledge that is available in all.
Professor Schweiger, please.
Framework.
Yes.
You have a specific research question about CLL, and I would like to use your data. Can I do this, or
Yes.
Do I have to go back to the patient?
No, we ask our patients to sign an informed consent form. This informed consent form has two different paragraphs. The first is, I want my chromosomes being investigated for my leukemia diagnosis. They say yes, because this is what they are asked to do. And then there's the next paragraph, and this is written, if there's any leftover material by anonymized data sets, I would also agree to support any research science within the framework of what is allowed in Germany. And when they say no, quite safe.
May I add a little bit regulatory stuff?
Yes.
I would suggest that you ask all your patients to be part of a registered trial located at your institution. And in addition, then a click to that you are allowed to share these data also with other institutions. I think this was your idea.
Mm-hmm.
And it's not really a broad consent, it's probably leukemia research.
Absolutely. And development of new drugs, for example.
In other words, there are three levels. One is the treatment trial, which you will use for your own publications on the topic.
Right.
And the sharing with others.
Yeah, thank you very much. This is a very good advice. I can tell you where we see the most problems. The problem is that the patient forgot to give him this paper to sign something. The patient would sign. Yeah. If we have a paper where it's yes or no, it's 99% yes. That's quite easy. They just don't have the paper to do this.
Yeah. But it has to be...
I understood. 3 levels, yeah.
Yeah, and it has to be separated.
Right.
for legal reasons also, because it's completely different if you ask for something for the treatment or for a specific study, and for something which you cannot describe yet, but the only thing you can say, it's leukemia.
Right, so you can,
research.
believe me, very expensive lawyers to help us to put this data there, and
For all who don't know, this is Professor Schmidt, by the way, the head of the ethics at the Technical
Yes.
University.
Thank you very much for this nice presentation. It's very enthusiastic, encouraging. I was wondering, it looks like everything is solved in the algorithm to get the precision you have, but how many cases? About
yeah,
this?
we have a, thank you for this question. We have a study. We have all consent, and this is gets my answer. And we now have enrolled 82 patients from all over Germany, and in 1/3 of them we have a new final answer that this is the disease and there is not nothing. So you can add with this information on the genome and the transcriptome for those very critical cases where we don't have any idea what's going on, for 1/3 a final answer and even a treatment after that. For the next bone marrow biopsy, because the problem is still there.
Do you find then a diagnosis which is among the genes and
Inflammation syndrome or something like that, that was not on our list.
Professor Haferlach, thank you very much for this impressive talk. Best view.
All right, it's me again. I'm sorry. I'll continue. And the question is, and I'd like to state or begin with a quote here. So the paradox of a clinical trial is that it is the best way to assess whether an intervention works, but it is arguably the worst way to assess who will benefit from it. And during the next slides, I'll try to elucidate this with you. All right, so there is a gap to fill. We have in our daily clinical duties all different shapes of patients every day. So they are from very old to very young, females, males, all different ethnicities. So this is the real world patient we treat every day. And if we think about randomized controlled trials, for a good reason, so like COPD are gone. Some ethnicities are not well covered in our randomized controlled trials. You see, the further we develop here, the more restrictive we are with our clinical trials. So what we try to do in our daily care is to treat based on evidence-based medicine. And this evidence-based medicine comes from randomized controlled trials. So this is the best kind of trial regarding the quality of evidence that we have. So I'd like to convince you that we might be interested in real-world data if used carefully. So data that derives from cohort studies, findings. So what is the potential that lies in real-world data? Of course, we can use randomized controlled trials and see if we can confirm those in all different kind of subpopulations. If this is true for example, the whole Bavarian population. We can identify unmet treatment needs. We can risk estimate the future cardiovascular event risk for individual persons. We can look if our patients take their pill. Guideline implementation. So there's a lot trials and patient treatment. Control trials, and this is the only use case I use today, so don't be afraid of all the others. It only takes 10 minutes today. We heard about, from the Google perspective, that this health insurance data or medical data is increasing day by day. So we are talking about zettabytes, and we heard how long it takes to download these movies. So this is unimaginable amount of data. We are not only looking in these interest in using this information from health insurance claims data, and this is a publication. This is from Dr. Crafting. He's in the audience today. I'm very glad to see him. This is a patient journey over time, and the different colors are labeling for different data sources. So the general practitioner, for example, the green dots, the red ones are the university hospitals. So we at the German Heart Center hold, for example, the acute myocardial infarction data of our patients, but that's only a very limited amount of data we have from these patients. But where all these data come together are the health insurances. So regarding that, there are legal and technical considerations that have to carefully to do care research. So we can improve risk prediction, we can improve the prevention, and we can improve treatment of these patients. So we partnered in DigiMed with the AOK Bayern. So this is the largest health insurer for general patients in all over Bavaria. And then you need a team of experts. So we have Dr. Krüger on the upper left, we have Dr. Crafting in the middle, myself, and then we have a lot of experience below. So Professor Castrati, Dr. Cassese, Professor Schunkert, that guide our analysis plans. So what kind of data do we have? We have individual anonymized data of 1.3 million individuals. The sex distribution is almost 1-to-1. We have a follow-up for all these patients over 10 years. And this longitudinal perspective, this is perfect. Regarding the cardiovascular, so on and so on. Randomized controlled trials and how to emulate those. So this publication is from 2019, and it was led by the German Heart Center in Munich. And what is the best? So we compared two. And what was shown is that prasugrel outperforms the risk of myocardial infarction, stroke, and death against ticagrelor. And this is encouraging. And what we wanted to see, if this is reproducible based on this health insurance data that we hold. But you should be careful. So if you emulate a trial, there are rules you should stick to. So think about a protocol and an analysis plan, publish those upfront. That might come from relevant factors that you did not think in the first place of. There should actually stop at this point, and so on. So you can separate I would give you an example about the limitations of matching. If you think about the NIH, this is King Charles III, and if you go with his covariates, for example, wealthy and famous. Okay, nearest number. Ozzy Osbourne. And arguably, they have so if it's a one-to-one, you don't have a problem. So a new field, the information of only two focuses on this slide. So regarding more than 20 perfectly to answer this question. We did individuals based on ticagrelor or prasugrel. Regarding the characteristics diagnosis at the beginning, unstable angina, non-ST elevation myocardial infarction, and ST elevation myocardial infarction, perfectly matched. And this is like the visual inspection. Matching is almost perfect. To the primary outcomes. So hold these RCT findings true in the general population or in larger data sets that derive from health insurers? The answer is yes. So we can replicate exactly the findings that were published in the ESA-REACT-5 trial. Prasugrel outperforms the in bleeding, so it does not come with a higher price of bleeding risk. For the secondary outcomes, I'm very quick with this. No significant difference for death, but prasugrel again outperformed this is crucial for our cardiovascular patients. So if we compare directly these randomized controlled trial to our real-world data trial, we have the hazard ratios on the Y and the X axis of the two respective trials, and you see the green line. And the closer you get to the green line with your findings, sorry, the better the overlap. So in this case, that's like perfectly overlapping. This randomized controlled trials, because we have more individuals we can look at, and even here in the Bavarian population, it's completely reproducible. So guiding personalized treatment, that's difficult as we hold anonymized data, but we can look and the acute myocardial infarction event, and these individuals to the it's easy because or more like so this woman 46 years old presented with an ST elevation hypertension and all vouch for prasugrel. So it's very likely that this patient goes very well with prasugrel. What you can see are the prescriptions for either prasugrel or ticagrelor over time between 2013 and 21. And the red line is when the publication of ESA-REACT-5 became available. And we learn for description prior and after ESA-REACT-5. So it basically means fear if you live remarks. Compliance in the broad randomized controlled trials have the potential to confirm, and that's important, to extend randomized controlled trials. Because, of course, we have the follow-up data of our study cohort for the next 3, 5, or even almost 10 years. And of ESA-RECT5 in the Bavarian population, and our data supports that most of the subgroups would profit from prasugrel in Bavaria, and measuring the reality of healthcare supply enables to understand individual and local needs for optimization of healthcare delivery aimed at maximizing the impact of therapeutics. And with this, I'd like to thank for your attention and the team who actually did the analysis, and this is Hannes Krafting in the audience and Niels Krüger online. Thank you very much.
Thank you very much for your talk. I have a question. So you started your talk with, you also said, the quote says that profit from it. Clinical trial with a large N. Well, who will benefit now from it? Like, how is this question addressed?
Yeah. Thank you very much. I did not say that we increased the population we look at by the now the power to look at these subgroups, and with ESA-RECT5 but we can now state that based on this data, specific subgroups would have a direct benefit out of changer, because no one would replicate this one-to-one comparison again. So the pharmaceutical industry is not interested. I see you have a follow-up question here.
Because you say subgroups, and do you mean subgroups, your characterization of the disease will benefit from the drug, this subgroup will get a disadvantage, or yeah, what do you mean with subgroup?
In this case, you are right. This is retrospective, only hypothesis generating. But it's not powered to own. But you would not have, or you would not be able to reach this power based on this patient population. But these subgroups most likely benefit from it. Good question.
Yeah, yeah. One more question. Is also notorious. Is a little bit you know.
Yeah, that's. It goes to clopidogrel.
Yeah.
So
Not-
there is
not
a
for-
rate. No. So there is at least not reported non-responding
Okay.
to pras. Patients that receive stenting are not able to metabolize clopidogrel, so they do not have any profit from it.
Okay.
And the question, of course, is: should we screen all our patients? So we can state at least for.
Okay.
And if we know that. Yeah.
Brilliant analysis. Thank you very much. Only in Germany, only in UK. and you couldn't do your view of enough patients. This is the advantage of Scandinavia, etc., over the Americans, and it would be great if we made that advantage more broadly available for research.
However, there is, for example, Professor Schneewer, from him and his group, but it's quite good with all. For example, the reimbursement for specific medications, this is kind of lousy.
You explained before with this HerzFit app,
Yeah.
that this is powered by the TK and that AOK is supporting. Can you explain a bit more the collaboration with these public insurers and what is the trade-off for you, for them? How is this defined? Thank you.
So what we like to bring in is the perspective of overall prevention. And there's no good funding in Germany for it. And I think the insurance companies learn that if we early on, we can save money over the long term. So I think that's their interest in us. So there is this kind of data for us, but it comes with the question, what can we do for improving the population? So stroke units, which needs more. This is a starting point, but basically have to Zentrum Gesundheit, where all this data should be available. Kind of a starter. We had the, I think, the potential to be a little bit ahead. Oh, so it's not only provided by the example, so there is no direct convinced that is helping. We did develop, or we helped to develop this app. We think it's good to use it. It's for free. But the owner of this app, for example, is the German Foundation or Heart Alliance or German Heart Foundation. And this is the patient perspective. So this is driven by the needs and interests of patients. So it's not in private hands.
Moritz, there would be one question from the internet.
Yes.
It's could you also evaluate the data from the other direction and ask what people who respond the opposite way have in common?
Yeah, excellent question. Yeah, of course. So I started with the highlight, but this is one of the key interests. And also to dissect what is different and may they profit from another kind of treatment. So I think we are very strong in metabolic treatment, like statins for the cholesterol metabolism, but we do not well yet on the inflammatory side, for example.
Another question, hooking into the remark with the Forschungsdatenzentrum Gesundheit. So the Forschungsdatenzentrum Gesundheit of the BfArM is overdue since 1 to 2 years to make the data available of all publicly insured people in Germany, which are like 73 million. So what you showed demonstrated the power such data have, even with such a limited data set. So my question goes into, shouldn't there be apart from single research projects, a very systematic and infrastructural approach to use these data to optimize guidelines and our health system?
The answer is yes. Short answer, yes. All right, time-wise, if there's no more questions, we would continue with the program. And I'm very happy to introduce Dr. Andreas Pichlmair. He's at the Technical University. He's at the Institute of Virology, and he worked before at the Max Planck Institute in Martinsried.
Very much for the invitation to speak here. Actually, I will talk about here in Munich and this regard, and it's called B3M. There are a number of proponents how do we deal with future pandemics and what is necessary to respond appropriately to. I said this is about to start, and we get very good. If we think about fighting a pandemic, there are 3 pillars that we think are important to fight a pandemic successfully in order to inhibit the collateral damage on society and also on economy. The first one is that there is a prevention of dissemination. And we know we went through the SARS-CoV-2 pandemic. There you can do lockdown. There's very little acceptance for this type of measures within the society, and it's also a severe economic burden. There's also vaccination to prevent virus diseases and spreads. We know that there is variable acceptance for that. The economic potential is very high, and both of these measures have been applied through the SARS-CoV-2 pandemic. It's still not very well really prevent not a epidemic or a pandemic disease is causing a severe disease in the patient. To epidemiology. Targeted and causal therapies, particularly when it comes to a single individual patient, of that, but there is still a lot of knowledge missing. The severity of the pathology Munich project, whereby we Helmholtz Munich different aspects that we are very interested in. The first one is to understand the pathogenicity from the viral side. What does the virus do in order to induce a severe disease? We also know that there is a host-directed disease progression. There's an immune reaction that is very often associated to a severity of disease. Comes together and methods and disease models. More specifically, in terms of viral pathogenicity, we want to ask what cells are infected, how do they that is very important is immunopathogenicity. As mentioned, the majority of diseases that we know are immune-driven diseases. There's a very high inflammation burden, and we still don't understand appropriately how this high inflammation burden is progression. And finally, we want to apply systems analysis. There's a lot of gravity here. Which are the cellular signaling networks that are affected, and how can this guide us for novel therapeutic diseases? So currently we are quite well equipped in understanding how viruses are interacting with humans, and very complicated if we involve drugs here, because drugs would affect the humans. They would also affect the viruses and even though we are very good in, as mentioned, to understand nowadays and classify how viruses are targeting viruses, we can basically we can identify drugs in order to prevent viral diseases. I just want to illustrate this in one little example here, whereby we, for instance, can infect cells, these are primary cells, with monkeypox virus. This was done last year during the monkeypox epidemics. And we can basically characterize the cellular responses by transcriptomic profiling, by proteomic profiling, and by phosphoproteomic profiling. And we get a large number of changes in these cells that tell us about dysregulated signaling cascades that are differentially regulated. So basically, we can identify virus-driven host pathogenesis and regulators that, for instance, lead to differential phosphorylation in cells. And basically, we try to use this type of this disease. Basically, in this case, for instance, we are using which are allowing us to integrate all the different data layers that I explained, and also basically add host-directed drug information based on Camber or drug bank, which basically allow us to shortlist drugs that could potentially affect the pathway. And through the functional screening platforms that we established here on site during the pandemics, we can also evaluate whether any of these drugs are really active against, in this case, for instance, monkeypox virus or not. And we basically can of monkeypox virus quite a number of candidates that were in part not yet known to regulate pox virus replication. Importantly, however, we are very conscious that we are not very good in that, because there's a lot of drugs that, for instance, we identify but that are not active. And one of the aims will be virus host interactions in order to make that better. And we basically used similar approaches and similar data, whereby we gathered data that will guide us towards a rational identification of drugs that could potentially regulate viruses. Prospectively, we think that we can integrate information on this drug virus host interactions and also integrate data from host biology and patient data that are actually coming from clinical trials that are ongoing here on site right now. And we would like to use AI to eventually tell us which are the driving factors that are driving a severe disease, and what are potential therapies that could, for instance, prevent disease progression. So basically, so far, as I said, this is a project that is just about to start. We got some we will establish a network based and we also have very strong connections to pharma and biotech corporations, which we think will be very beneficial in case of a future pandemic to be in place once such a pandemic or epidemic strikes. Currently, one of the bottlenecks we are still working on, and for which I think this meeting will also be very fruitful for, is to solve a few issues that we still have on storage capacity and computational infrastructure. So prospectively, we would like will use and settings and use AI-based analysis for multi-omic studies in order to prevent pandemic-associated pathologies. Importantly, this will be done by 3 main research institutions here in Munich, and also together with companies that we already teamed up with. And a very important partner here is also Roche, who will basically counterfinance part of the project that we had proposed here. And finally, as said, these are the proponents at this stage. We are very much looking forward to a very exciting project that will run for the next years. Thank you very much.
Professor Pichlmair, thank you very much. Questions? Don't be shy. Or I pick one.
Thank you for your talk, first of all. There, because I saw that it only includes 17,500 genes. Yeah.
Well, there are different omics layers that we will use. When we talk about interactomes, it's more protein-protein interactions that we are talking about.
Which one?
We will have a priority system here for certain diseases that we feel are important for pandemic and epidemic progressions. One is SARS-CoV-2, because we have a lot of information on that already, and we can basically gather and use this data to interpret it. Another one will be influenza virus, and a third one, which is actually a bit neglected, but is very important, is priority programs that we will work on in this case. The interactomes themselves are not as rich, so they are not 17,000 genes. So very often these are more in the hundreds or thousands, which are, of course, complemented by additional data based on transcriptome, proteome, or phosphorylation, or PTMs in general in these cases.
Thank you.
Your chance. No more questions?
Thank you very much. Data and compute infrastructures. So first question is, why don't you simply go to AWS, as Professor Haferlach showed? And the second question is, what is your best approach now to that, to solve this problem?
As said, we are still setting this up. We are not there yet to have solid, concrete solutions. We have a lot of clinical data that would be integrated here from patients. So this is a bit of an issue, of course, to go to non.
Professor Pichlmair, thank you very, very much. I see you are looking all forward to lunch, but it's not yet. I'm very happy to introduce Professor Schmidt. He's the head of the ethics committee locally at the Technical University in Munich, but he's also overlooking all the ethical committees all over Germany. Yeah. So we're very much looking forward to your talk. Thank you.
Yeah, thank you very much. I would like to add that my working group, my scientific working group, digitization and large amounts of data is daily life for me for many, many years. But here, first, I'm happy that I was invited. It's an honor for me. And second, the organizers gave me a nasty title, namely, is data-driven medicine a benefit to society or an empty promise? And I will try to give a fair, I would say, from a more distant perspective. As the chair of the ethics committee in this university, I see more than 800 applications a year, and an increasing number of applications are dealing somehow with artificial intelligence, not necessarily on a high quality level. Therefore, we included 5 new members, specialists for AI in order to find out what is behind all these approaches. I would like to start with an outline of the difference between hypothesis-driven and data-driven research. In the hypothesis-driven research, it is common to develop an idea or an assumption and this, which then has to be tested in a carefully designed prospective study. Expected effect strength has to be considered, and the number of subjects included have to be calculated, and at the end we can test the hypothesis, reject it or not, and can have a conclusion. The data-driven approach eliminates the need for predefined hypotheses. Instead, artificial intelligence, such as machine learning, are used to search for irregular patterns, and these findings must then be interpreted. We use usually some kind of data mining techniques are used, like with all kinds of AI technologies. And we get an explorative finding, and eventually this explorative finding might be the starting point for formulating a new hypothesis. Will be the start of a confirmatory study. AI is playing an increasingly important role in recognizing patterns. At this, which is why we like search images like this. I don't know how long you would need to find all the 10 or 12 errors hidden there. In this case, and this is a very small section of the human genome from 2 individuals, detection errors in the base sequence with the naked eye is far beyond our capabilities. However, even in case of whole genome sequences, a computer equipped with appropriate software can identify the points of interest. This is imaging, pattern recognition, but there is another problem. Pathophysiological pathways are functional changes on a molecular level that are associated with a particular disease. Living organisms are very complex. Information flows at multiple levels. The omics we heard about so quite often today. With genomics representing, of course, the genetic information, and phenomics representing conventional clinical findings, which nowadays often is being supplemented by telemedical data. The discovery of new pathophysiological pathways through AI techniques is very difficult, but it would pave the way probably for the development of new diagnostic tests and therapeutic approaches. You see, this would produce some breakthroughs eureka moments in medicine, and up to now we spoke about speeding up clinical processes by these assisting techniques. But I think this is possible, might be possible, but it's much more difficult to achieve. If high-quality data are collected in a structured manner, stored in large data archives, and made available to researchers. In Germany, extensive groundwork has already been done. I would like to mention 3 of them. On the left here, you see a map showing the activity or the locations of the Medical Informatics Initiative, which is funded by the German Federal Ministry of Education and Research, and in which almost all university hospitals are participating. This system provides archives for decentralized data. In contrast, and we heard of this idea also 2 or 3 times already today, the German Federal Ministry of Health and Research, 2 different ministries have different ideas, is planning a central national data access and coordination center at a higher federal authority, the BfArM. And third, we heard this also this term, and I understand this as a virtual space in which all types of decentralized data can be temporarily consolidated and analyzed in a secure environment. And as you see, these ideas are completely different, and do not trust this solution. Yeah, because then you have all this is the place where new drugs are certified for the market. On the other hand, here you have all the health company data, and in future you will have a lot of the so-called elektronische Patientenakte, electronic patient form or patient report, and they plan also to add scientific data from clinical trials or performed by industry. And then you have all the information there, and the newest information I got is that they plan even a central ethics committee. And I believe that this is not such a good idea. I would like to combine these. A secure cloud, a secure space where you can put data, make some analysis, and then take them out again, and you can take data of any origin. Maybe a combination of both would be a good solution. So far, the results, we heard a lot of optimistic things today. The key lecture showed us a lot of future fairy tales, I would say, and the possibilities. And if you look into the literature, the situation is much more difficult. The results of medical AI research so far have been mixed. There has recently been an enormous increase in the number of publications, inducing, by the way, a publication bias, because it's quite simple to get a paper published into a journal, because the journal knows there's a search is starting, and the papers will be recited with high probability. We saw that in the COVID pandemia also, where pandemia paper were published a lot and recited. However, many of these studies are small and exploratory and often lack external validation. The other day, 3 days ago, I was with the German Ministry of Health in a retreat where AI researchers were met. They had in common that they were funded by the Ministry of Health, and they exchanged their information, and I was a little bit surprised about the low quality of most of the papers presented there. This is a list of papers published in high-ranked journals, and it's just a collection of the conclusions, some words, and there's a lot of conjunctive wording: has the potential, might have prognostic value, holds important promise, could provide, we believe, can lead, could be part, other studies should be. You see, this is the situation, and therefore we definitely need standards for the design of protocols and standards for the publications of papers. This is, this is such a the consortium. This was the consortium itself is 12, 15 years old, and it was complemented by AI rules. And these some of the important AI rules state that in the paper, the criteria for inclusion and exclusion for the participants and input data, that's very important, should be stated. The AI algorithm version used should be stated. How the input data were taken from. And please explain how data of poor quality or missing data, input data is handled, and detail the interventions output. And you see, this is very recent development. This paper, I think, is 2 years old. Yeah, it's from 2020. And here I give fixed, or which, and as you can see, it was a randomized, controlled, population-based trial, 80,000 women, aged 40 to 80 years, eligible for mammography screening at sites in Sweden. And AI-supported screening was the intervention group, and the standard group had just a double reading by radiologists without AI. And interestingly, the AI-supported mammography screening resulted in a similar cancer detection rate, double reading, screening reading workload. I think this is very similar. The AI, not worse, similarly good as the expert, but it saves a lot of time. And this time, of course, we should give the use for the interaction with the patient care. I think this is definitely true. Therefore, the first answer, societal value, is of course there. There's no doubt. This is some kind of summary. AI is particularly successful at identifying complex, surpassing, especially in imaging and genetic data analysis and all these omics analysis, and high-quality data collection and analysis infrastructure, like Germany's Medical Informatics Initiative. It's a really good idea, and it's would be a good partner for this kind of stuff. And so far, the studies show potential, but lack extensive validation. This is one point. High-quality studies demonstrate AI's effectiveness in assisting diagnosis, but I would like to add the real breakthroughs, the identification of so far unknown pathophysiologic mechanisms, which could be the starting point for new diagnostic tests or new therapies, new targets for therapies. These breakthroughs, I didn't see them in the literature yet, but I very much hope that they will come in future. And as said, the reproducibility validation of AI models in independent data sets is necessary and will hopefully overcome the reproducibility crisis, which we have. At the meeting which I mentioned, models which worked in Berlin very well didn't work at all in Cologne. And that's the problem. And all these little studies are published, and there is a huge stack of hay and with only very rare needles in it. Thank you very much.
Professor Schmidt, thank you very much for these important insights. Are there questions? I'd like to start with one. Yeah. So regarding your very initial sentences, that you receive applications and the young investigators and the old too are interested in implementing an AI in the research. And you mentioned that you recruited 5 more persons that are experts in the field, or members.
Members of the ethics committee. Yeah.
And my question is, do you also provide, so you
we are doing, I'm personally doing, together with these experts and the 5 to 10 video conferences a week, each of lasting 30 minutes, in order to find out where the problem is, and very often the statistician also is involved then. So we can, yeah, I think we are acting as an internal review board of the university. That's I should maybe explain. I'm doing this job in the following way. At first I try to find out, is the question make it sense? If it does not make sense, the question, the study is unethical, without any doubt. The second question is the methodology appropriate? Can, with this technique, with the design, can the question be answered? If this is not the case, it's unethical. And the next step, of course, safety and security of the patient, and a fair, informed consent process. These are the three most important things we are looking at. And we try to be flexible. You see, the world is changing all the time, and AI is now a big surge. Therefore, we had no chance to do it, not to do it.
Thank you. Professor Schmidt?
Last question before lunch. AI is actually the older term. It has been brought up in the 60s. And then for many, many years, people were using the more rational term, machine learning. And I would really hope that people would rather use this term than this buzzword AI.
Yeah.
And realistically, except for this image analysis, very impressive and so forth, I think it's too early for machine learning, for precision medicine. First, we need dozens of cases from which these machine learning algorithms can learn. Then we need another dozens of cases which these algorithms correctly predict, and then we can let them loose. So I think it will come, but it's totally oversold in the moment. It's just too early, with the exception for image analysis. There it works. Yeah. But to subclassify patients, predict drug treatments and so forth, it's too early. Yeah. Everything we do, let AI jump on it. I mean, you know, bad data in, bad data out. It will be important, but it's too early.
Yeah, thank you very much. I would like to support your opinion. Maybe the term. There is one term which I like even more. It's smart algorithms. It's statistics in the end with very smart algorithms, and I like statistics, and I was very surprised how smart these algorithms really are. So that within one big data set you can, but it's still no, and that is my message to all the younger persons in the room. Is a buzzword, everybody does it, but be careful what you are doing. And yeah, maybe I end with one saying which is common now between the members of the ethics committee. It is not directly linked to smart algorithms, but with apps. We call it jeder Depp schreibt eine App. Yeah, there are thousands, and it's just on a variable. This is not that doesn't make much sense. But sorry for that.
No, thank you very much for the perspective of the older generation. So next session B at 1 o'clock. Thank you very much. Enjoy lunch.